Hi. For those of you who don't know me, my name is Bridget, and I'm a senior. And this past summer, I participated in the Discovery to Cure research program at Yale. So I worked for 6 weeks in a cancer research lab in their therapeutic radiology department. And the lab focused on drug discovery and high-throughput screening for inhibitors of N-link glycosylation in cancerous cells. Okay. So firstly, just some background. Proteins are made in a place called the endoplasmic reticulum, or the ER, in cells. And the ER is really important for our research because that's just where it was all centered. And after some proteins are synthesized, they require further modifications. And glycosylation is just one of those modifications. And it involves the addition of a protein and a carbohydrate, or a sugar. And so N-link glycosylation is just a specific kind of glycosylation, but it's really important because it occurs in the endoplasmic reticulum, and it involves lots of different steps that are catalyzed by different enzymes. And that's what that big picture is showing. That's kind of overwhelming, but it's just so you get a sense of how complicated the synthesis of N-glycans is. N-link glycosylation is important to ensure proper protein folding and maturation, and thus aids protein transport through the endoplasmic reticulum, or the ER. Disruption of NLG, or N-link glycosylation, interrupts ER homeostasis, initiates the unfolded protein response, and if left unresolved, will lead to cell death. So the research that I did this summer was centered on 2 novel drugs that impact the synthesis of N-glycans and how that impairs cell function in different cancer cell lines. So the unfolded protein response, or the UPR, is activated in response to an accumulation of misfolded or unfolded proteins in the endoplasmic reticulum. In this case, the UPR has 2 primary aims, which is initially to restore normal function of the cell by halting protein synthesis and activating signaling pathways, which will lead to the production of more molecular chaperone proteins. Chaperone proteins are specific kinds of proteins that are designed to help correct misfolded proteins. It's really important that proteins are correctly folded because protein shape is really important for their function, and so the proteins won't be able to function if they're not shaped correctly. Binding immunoglobulin protein, or BIP, and calreticulin are the 2 main kinds of proteins that I screened for this summer, because if we see an increase in either of them, then the ER is in stress due to the presence of misfolded proteins. BIP is a protein that's recruited during the unfolded protein response because it helps the cell regain control of protein synthesis, and in cases when it can't correct the misfolding, then it'll target the proteins for degradation in the ER. So therefore, an increase in BIP suggests that the ER is stressed due to the unfolded protein response because the unfolded protein response has been activated. So if we see more BIP, then it can be inferred that these drugs are disrupting normal cellular processes. And then calreticulin is just another type of chaperone protein, and it works in a family of chaperone proteins, which ensures that only correctly folded proteins are transported out of the endoplasmic reticulum. So therefore, the same logic that we use when screening for BIP is applied also to calreticulin. And there's another family of proteins called receptor tyrosine kinase, or RTK proteins, and they have an increased presence in cancerous cells, and their upregulation promotes the growth of malignant tumors. So previous research shows that inhibition of N-link glycosylation reduces protein levels of these overexpressed RTKs. As a result, RTK signaling is reduced, suggesting that inhibition of NLG is a possible approach for targeting RTK signaling in tumors. This flowchart just shows the steps in a high-throughput screen that my lab did before I arrived. They were looking to identify small molecules that interfere with N-link glycosylation because that would consequently impair the signaling initiated by RTKs in tumors. And from the screen, the lab found 2 novel drugs, or compounds, which they called NGI-2, and that's N-glycan inhibitor. Okay. So the main focus of my research was on these 2 drugs because they're still quite novel, and the lab is in the process of trying to learn as much as possible about their different functions and at what concentrations they're most effective at and at what times they have the greatest impact in different cell lines and cancer types. So the hypothesis is that these drugs inhibit N-link glycosylation, which should eventually lead to a buildup of misfolded proteins, causing ER stress and ultimately causing cell death. Cell death would be good in this case because they're looking to kill the cancerous cells. So this summer I worked with these 2 drugs, and I added them to different cell lines and assessed them in cells that had been treated with different concentrations of them and at different times. My objective was to see that these drugs are interrupting NLG, which causes a buildup of misfolded proteins in the ER, and in turn stimulates the unfolded protein response. So I used 2 human multiple myeloma cell lines, ARK and ARP, to study this summer. Multiple myeloma is just a specific kind of cancer, and it's generally associated with the bone marrow because it impacts immune cells, and immune cells are found in bone marrow, generally speaking. These tumor cells are especially useful for our research because their ER is highly developed, and that serves to maintain equilibrium under a condition of excessive ER protein production, which makes them especially useful to us, seeing as the research I did this summer was centered around the ER and causing ER stress. So the main type of experiment that I learned how to do was called a Western blot. And Western blot is a widely accepted analytical technique that's used to detect specific proteins in a given sample of tissue extract. It uses gel electrophoresis to separate denatured proteins by length, with the shortest proteins traveling the farthest down the gel. The proteins are then transferred to a membrane where they're stained with antibodies specific to the target protein. After exposing the contents of the membrane on a piece of film, the blots were a great help to demonstrate the differences in the protein synthesis between my control groups and the groups that had been added, that had had drugs added to them. So the first line here with the 0 above it is our control lane, and those are the ARK cells that had been treated with no drugs, so they're just normal myeloma cells. And then the second 2 lanes are cells that have been treated with the NGI2 drug for 24 hours in different concentrations. And the 4th line is our positive control, and it is the same cell line, but it had been treated for 24 hours with tunicamycin. Tunicamycin is a well-known N-glycan inhibitor, and so we wanted to use it in order to see how similar the protein synthesis after our drugs was compared to tunicamycin, because it should be somewhat similar. So after running the Western and performing the binding with antibodies, these are the results I found. So we can see an increase in protein is also depicted by a thicker, darker band, which you can see. So we can see an increase in the amount of BIP and calreticulin, which are those chaperone proteins, in the lanes that have been treated with the NGI2 drug compared to the control. And there are also results similar to the 4th lane, which is the tunicamycin. So that's good because that's what we were looking to see, because we want to see that the ER is in stress, and so these chaperone proteins have an increased presence. The actin blot on the bottom is used as a loading control, and we use that in order to validate the differences that we see between the other results, because actin is a structural protein, and its synthesis should be independent of N-glycans, so therefore it should be the same amount in each of the cells, and that just tells us that we're not getting a false positive with the differences that we see. So therefore, the results for BIP and calreticulin demonstrate that there was an increase in the synthesis of both chaperone proteins in the cells treated with the NGI2, meaning that the drug did cause ER stress, ultimately inducing the unfolded protein response. So based on my research, we can see that by inhibiting N-glycosylation, the drug NGI2 generated ER stress and ultimately induced the unfolded protein response in the cells because we saw an increase in those chaperone proteins. In terms of further research, there's still a lot to do because the drugs are still hard to completely understand because we don't know where exactly they act in that, I don't know if you remember, kind of complicated diagram I showed on the first slide, because we have to pinpoint more exactly where the drug behaves in the process of synthesizing N-glycans, and that'll help better predict how they behave in different types of cell lines, because right now we're only focusing on certain cell lines that we already know a lot about. And it'll also be important to test the use of these drugs with other drugs that are being used for cancer treatment, like bortezomib, to see the possible benefits that their joint effect could have on cancerous cells. Bortezomib is a drug that is used currently in the treatment of cancer, and it impairs the function of proteasomes in protein degradation. So theoretically, if we combine that with our NGI2 inhibitor, we could expedite the induction of cell death because there would be a greater accumulation of misfolded proteins in the ER. So we can see that there is a lot of research that still needs to be done, but it's pretty exciting, and I'll look forward to seeing what the lab does in the future. So that's it.